Okay, everybody, so in this video I'm going to be going over the MCQ progress check for Unit 1. Now, this is going to take a little bit longer because there's more questions, so feel free to hop around and look at the specific questions you felt like you needed help with. I do want to start on this page, though, which is the sort of score breakdown by topic, 1.1 through 1.6. And then the sort of class average. And this tells me right away that based on this, the thing that as a whole class, you all are the weakest on, the thing you need to spend the most time reviewing is the structure of water and hydrogen bonding. So you need to be able to just be solid on covalent bonds versus hydrogen bonds, which one includes shared electrons, the arrangement of individual elements in a water molecule, and then the arrangement of water molecules relative to each other in solid, liquid, and gas form. You need to be able to connect hydrogen bonding to the properties of water at different stages and things like that. It looks like water is a weakness. And I don't know if it's just because that's the one we did the farthest to go or what, but make sure you're going back and reviewing those videos. Bozeman Science has a bunch of good videos that I might put up that you could use to supplement. And you can always extend if you have questions, ask me questions. So let's get started looking at the questions themselves. So these are not in any particular order. Question number 1 here. These are in the order that I have them on my page. I'm not sure if you got these questions delivered to you in the exact same order. So, but question number 1 shows some DNA molecules. It asks which feature of the model best illustrates how biological information is coded. And most of you selected the correct answer that it's the sequence of the base pairs. Some of you selected that the 5' to 3' end, which is a feature of nucleic acids, may be involved, but that does not determine the information storage. It only determines the directionality, and then the lines connecting sugar and phosphate that represent covalent bonds. Again, that's a true statement. Those do represent covalent bonds. However, it does not carry the biological information that's coded in DNA. Question number 2, we stay with that same molecule, but they add an extra diagram here. So it says 4 different bonds, W, X, Y, and Z in a DNA molecule. This one looks like it was confusing for a lot of you. It may be helpful to make flashcards or to create a foldable template to do something other than just read it and then reread it and then reread it. Some kind of like quizzing thing about the different bond types, because I get the sense that this is a weakness for some of you. So covalent bonds versus hydrogen bonding, how they're demonstrated. The correct answer, the most often selected answer, was bonds Y and Z are the same. Well, let's see. What's the question, though? To repair some types of damage, a single nucleotide can be removed from a DNA molecule and replaced with an undamaged nucleotide. Which of the 4 labeled bonds in Figure 1 could be broken and removed to replace the cytosine nucleotide without affecting the biological information? Okay, so there's only one cytosine nucleotide here, so you need to find this one. And then you need to know how DNA is put together. It's put together with a series of nucleic acids, and a nucleic acid contains a sugar, a phosphate, and then one base. Now, you need to be able to look at the directionality of these molecules, but the only way in this diagram that you could cut out the cytosine would be to make a cut here and a cut here. Now, some people might get stuck and they might get tricked and be like, well, why wouldn't you just cut it here and then up here and give it this phosphate? Well, it does matter which phosphate goes with the base or with the rest of the nucleic acid. So you have to read it from the 3' to the 5'. So the phosphate is going to be carried with the 5' end of so the 5' end is here, so this phosphate belongs to this nucleic acid. The 5' end is here, so this phosphate belongs to this nucleic acid, and so on. In either case, some of you said bonds W and Z at the same time, so if you cut W and cut Z, that would remove this whole line, this whole layer here, but would not allow the cytosine to be cut out, because the cytosine would still be contained within the DNA double helix. The key here is it says without affecting the biological information. If you cut here and here, you're going to remove a T and an A, and that would change the biological information. Whereas if you just cut this C out and put a new C in, it won't. Okay. This next one, another one, looks like most of you are pretty comfortable with. A student wants to modify model 1 so it represents an RNA double helix instead of a DNA double helix. Of the following possible changes, which could be most effective in making model 1 look more like RNA and DNA? So changing the deoxyribose to riboses and adding OH groups would do that. It's the... or by adding. The addition of OH groups change a deoxy. This is an oxy group. So deoxyribose in DNA is missing an OH group. Some of you got distracted by changing the sugar phosphate to a ribbon. No, that has nothing to do with the shape of the molecule. That's simply an artist's diagram of it. Or change the sequence of the base pairs. Again, that doesn't... Changing the sequence won't change the type. Now, if you replaced the thiamines with uracils, that would be part of changing it to a RNA molecule, but not changing the sequence, right? Changing the type would change it. Now, if that option said there change the thiamines to uracils, then you wouldn't be able to tell whether B or that one was correct. Those would both be correct then. But in that scenario, only B is a correct answer. Okay, so next we have a question about starch and cellulose. It looks like most of you did pretty well on this one. The question is about starch and cellulose that's repeating glucose monomers, something you should understand, but that in cellulose every other glucose monomer is rotated 180 degrees, and though in every other way they're the same, that rotation completely changes the behavior of those molecules. Cellulose is what makes up wood. Humans can't digest cellulose. Starch is what you find in a potato or rice, and we can digest that very easily. But in every other way, it's identical. We look at that and we're like, what? How could that make that much difference? It does, because when you have to chemically separate these, the enzyme in your digestive system that's going to break these apart can't interact with cellulose the same way it'll interact with starch, because the starch molecule is shaped differently, so the shape of the protein won't fit into cellulose. Okay, but you guys did pretty well on that one, understanding that shape determines function. Same deal here, basically the same question, again talking about the differences, and it's these assembly and organization of monomers in a polymer results in chemical properties. That's a fundamental idea. It looks like you guys have a pretty good handle on that. Dehydration synthesis, this one tricked a couple of you. The question asks which of the following describes the process that adds a monosaccharide to an existing polysaccharide. That means that would add one more of these monosaccharides to the growing chain. To do that, we're going to have to remove a hydrogen and an OH group off of, there'll be a new polysaccharide or monosaccharide that'll come in. An OH group will pop off of one end. It'll knock a hydrogen off the other end, and then that'll form a bond there between the oxygen, and that will release water. A specific enzyme removes the hydrogen from a monosaccharide and the hydroxide, the OH group, from the polysaccharide, creating a bond between the two and creating a water molecule. So, ionic bonds not really involved here. A monosaccharide is completely broken down. No, the monosaccharide mostly remains intact. It's only a small piece of it that is involved in the dehydration reaction. And that's dehydration synthesis is something you need to understand. So make sure you are reviewing that. Number 7, different polysaccharides are used by different plants. So same theme here. Which of the following could most likely occur if cattle lost the ability to maintain a colony of microorganisms in their digestive tract? And most of you selected the correct answer here. Cattle would no longer be able to digest cellulose. And that's what this reaction shows here. That enzyme B can digest the glucose, or can digest the cellulose to produce glucose. However, it cannot, interestingly enough, digest starch. So the microorganism creates a specific enzyme for cellulose, but not for starch. You guys did pretty well on that one. Ice on a pond. This one stuck. Got a lot of you stuck. Which of the following describes how the structure of ice benefits these organisms? And I think this same concept caught you on the practice quiz on this. Water does not become closer together when it freezes. It does in almost every other kind of molecule that exists except water. So water is unique and special because when it forms ice, the water molecules actually snap into this crystal structure, which is less dense. They're not moving as much, but they are less dense than when they are as a liquid. Okay, and that does form, allow the warmer, denser water to stay liquid underneath. A lot of you got tricked by B. And this second part of the question is true. Ice forms a barrier that protects the organisms in the water from freezing air temperatures. That's true. But the reason for why water molecules in ice are closer together, they are not. They are farther apart. So make sure you go back and review the structure of water and ice. Again, this one, a lot of you got this one wrong. A lot more than I would have expected. And I think, and it's spread out over all of them, which tells me that people who are getting this wrong, and maybe even some of you who are getting this right, just happen to guess the right one, do not understand how two different water molecules are related to each other, where that bond is. I think you may have gotten stuck here by this negative, I think, but that's not supposed to show a negative charge. That's showing a bond, the hydrogen bond. So again, go review bonding if you got this question wrong. For water specifically, be able to draw it out. Where's the covalent bonds? Where do the electrons go? Which is the more electronegative, which is the less electronegative element in water? And then, what is the polarity? Where's the positive end? Where's the negative end? How do they attract to each other? Question 10, transpiration. A lot of you got this one wrong too. And this is the last one I'm going to do on this, and I'm going to have to make a separate video for the rest of the questions. This is just testing your understanding of adhesion and cohesion. We see that we have water moving from the soil up through the tissue of the plant, and then evaporation of water from the leaf. So water molecules are moving along these xylem tubes, and we see a vertical movement. I think those little dots each represent water. And then the water leaves from the leaf surface. So what is happening here? Well, the water is attracted to the water. So as the water leaves the leaf, it's going to be pulling on the water underneath it. Okay? Water doesn't push on other water, it pulls on other water. So the correct answer is that as water exits the leaf, hydrogen bonding between water molecules pulls water up from below.